I think you can hear me in the back. Yeah. Right.
Here's a stick.
Right.
And we'll sign it for 5 minutes.
Okay.
Before you.
Thanks very much.
The next stick is.
Well, I just asked that and she said not. But I don't know if that's true. Is the microphone for the recording or is it for so that you can hear me?
The recorders want you to use the microphone.
Okay. All right. I'll try it.
I hate this. If I have to do it.
That's fine. I'll give it a try. Oh, so I just hang on to this thing.
That's, you put it in your pocket.
All right.
So this goes somewhere on your,
I've never used one of these.
on the red thing.
Yeah.
This just goes somewhere in your pocket. Yeah.
That works. All right, great. So I'm Gavin Naylor from Florida, but I perhaps don't sound like it, but I am. So there's been a long history of interest in estimating the relationships among the major groups of animals. And this goes back probably most notably to Haeckel in the late 19th century. But there were other people too. Bateson, at about the same time, was interested in the origin of chordates. And Cope, too, was interested in the relationships among major groups. And what's somewhat surprising is there wasn't really much interest after this initial interest until late in the 20th century, notwithstanding the work of Libby Hyman, who looked at the relationships among invertebrates. And perhaps this was because there weren't really many characters that they could homologize between worms and vertebrates, for example. But when the molecular phylogenetics came along, people went nuts. And there was this really influential paper in 1988 by Field looking at 18S ribosomal RNA and comparing the relationships among different groups of animals. And this spawned tremendous enthusiasm. Within about 10 years, there are about 100 papers looking at the relationships based on 18S ribosomal RNA. And everybody thought this was going to solve all of their problems. We'd know everything about all kinds of relationships. And what was happening as the consensus grew is as more and more trees came forward, they found out that, my goodness, there was no resolution at the base, but there was a lot of resolution at the tips. Well, clearly, this was quite consistent with a Cambrian explosion. So people started to explain, well, 18S is actually reflecting the signal of a Cambrian explosion. But then a paper was published by Abu Hayf et al. About 10 years later, so only 10 years after the enthusiasm for this new method came about, we see that somebody goes and pours water all over the idea and says, well, actually, maybe the marker isn't as good as you think, and it's not a question of it's reflecting the truth, but the resolution is a shortcoming of the marker. Well, biologists are furiously optimistic, and whenever they can't get a good answer, what they do is just collect more information. So obviously, it was just a problem that we didn't have enough information. And so at about the same time, in the mid-1990s, some people were more arduously collecting whole mitochondrial genomes. And this was really quite a good way to estimate phylogeny because, first of all, you could get a lot of information fairly easily. It was easy to collect. There's no introns, and compared to nuclear data. And then the mode of inheritance was fairly well understood. And really importantly, we didn't have these problems of paralogous comparisons. Orthology was fairly reliable and easy to ascertain. But people did start to notice that even with a lot of information, we'd start to see some quite controversial and peculiar groupings coming out. And different mitochondrial genes often suggested conflicting inferences. So I was sort of curious about this, and when I was a postdoc 10 years ago, and Wes Brown said, have a look at this Amphioxus sequence and do something interesting with it. And that's really exactly what he told me. So I thought I'd collect all of these mitochondrial genomes together for groups of organisms whose relationships were really well understood, that were uncontroversial. So here's the tree for these uncontroversial relationships, and it's really obvious to you, of course, what's going on here. Now I should put some labels on it for you. But there was a bunch of mammals, and outside mammals, we had a bird. This is 10 years ago. We didn't have that many sequences. You have to be forgiving. We had the frog. We had a couple of fishes. And then we had a lamprey. And we had Amphioxus, or this cephalocordate at the base of the chordate group, And then outside of them, an outgroup of deuterostomes. We had a couple of sea urchins, and then some very distant protostome outgroups. When we subjected this to phylogenetic analysis, some parts of the tree seemed very reasonable, but some were missing. We had this very strange grouping, which appears to have gone away. That's really peculiar. The host doesn't like it. Oh, there we go. Where chicken, frog, and the fishes clustered together, and even more peculiarly, sea urchins came inside cephalochordates. So we've got sea urchins, or lancelet, the Amphioxus, at the base of chordates is actually outside of sea urchins. Well, this could be just some sort of statistical slight anomaly, but what happened is this placement of having Amphioxus outside the echinoderms reverberated at the level of nucleotides and of transversions and of amino acids, at least under parsimony. And it's very well supported by very, very strong bootstrap support. So I thought, well, it's obviously just some sort of parsimony issue. So I thought, well, have a look at them, the nucleotides at least, under likelihood. So I looked at all 16 canonical likelihood models at the time. So we've got Duke's Cantor, cumulative parameter, HKY, and GTR across 4 different rate variation schemes. And every single one of them failed to give the expected tree. All of them put echinoderms inside of cephalochordates. So some people might think, well, your expected tree is wrong. But I was sort of persistent. I was quite sure that comparative anatomy, and there's quite a lot of evidence to suggest that cephalochordates are indeed the basal group to the rest of the chordates. I assumed the results were misleading. So I thought, well, if they're misleading, what's misleading it? So I just forced the sequences and interpreted them on the expected tree and had a look, tested the fit of different classes of information. I had a look at which genes had a good fit or had a poor fit, which codon positions, which amino acids, chemical properties, charge, hydrophobicity. And you can see here on the y-axis is, as you go up the y-axis, is a better fit. And you can see very clearly that third codon positions have a poor fit. Everybody's used to that. But you can also see that certain genes have better fits than others, certain kinds of amino acids, certain charges, and certain hydrophobicities. Well, to sort of quickly summarize, what we found out is that the subset of nucleotide sites that coded for proline, cysteine, asparagine, methionine, and glutamine yielded the expected tree, and the hydrophobic residues, isoleucine, leucine, and valine, were found to be especially misleading. So we concluded, unlike most biologists, they didn't need a lot of information, that it was actually more important to tailor the models to accommodate structural and functional constraints rather than just collect more and more information in the hope that somehow the truth would be revealed in the data like the face of Jesus. Notice I didn't say the face of Muhammad. So at that time, we weren't able to do amino acid likelihood because the computationally it was sort of tricky. But more recently, Dave Swafford has actually incorporated likelihood models into PALP, and we applied the MT-Rev model to the amino acid transformation of exactly that same data set. And what we found, quite appealingly, is that we get a much, much better tree, except for the fact that the frog still clusters with the fishes, and it does so with really strong bootstrap support. But if you add in a gamma parameter, then rather appealingly, we get exactly the expected tree, and we do so with really strong support. All this goes to say that modeling the substitution process appropriately is critically important. Remember that all of the canonical models at the nucleotide level gave the wrong inference, and quite emphatically so. So the fact that we get strong support for all of the nodes for an incorrect topology with the frogs and the fishes coming together is somewhat sobering. It underscores that when you don't include the gamma parameter, that the bootstrap support reflects the sampling variance of the signal induced from the interaction between the data and the model, not a reflection of phylogenetic accuracy. For the inference to be accurate, it's got to be unbiased with respect to the process that gave rise to the data. So we have to think of this as a sort of landscape of for a given combination of organisms and their sequences, as the sequence length increases with the same character patterns, so the topography of the peaks and valleys they're approximately the same, but they get vertically exaggerated, resulting in a more decisive landscape, but with lower sampling variance. So as you change the model parameters, you sort of have tectonic shifts in the landscape, and some peaks go up and some peaks go down. But the model has a lot of influence. So the bottom line is the details of the model are critically important, especially when you apply it to really long sequences and a sparse sample of very divergent taxa. For these situations, you get very little help from the data itself to estimate the patterns of change. Most of the estimate comes from the model itself. Well, think about this. Very, very long sequences, very, very sparse taxon sampling. It's exactly what these phylogenomic studies are. So there was a really nice phylogenomic study done by Frederick Delsuc in 2006, and they had 146 EST genes, much, much longer than anything I looked at, for 38 taxa, ranging all the way from fungi to vertebrates. And when they subjected the data to analysis, they actually found out that cephalochordates, Amphioxus, was a really special kind of sea urchin. So it clusters with sea urchins, and these guys did a really great job. It was very thorough. And so they published a paper in Nature saying tunicates and not cephalochordates are the closest living relatives to vertebrates. So sea squirts were basically chordates, or the closest living relative to vertebrates, and cephalochordates were outside. Now, these guys did a great job. This was not just fly-by-night. They're very, very thorough. They used all kinds of different analyses, partitioned likelihood, individual analysis for all of the 146 genes, and they've got a fairly emphatic result. Tunicates as the sister to vertebrates and Amphioxus with the sea urchins. Furthermore, they went on to look at competing topologies and showed that the competing topologies under these models were statistically significantly a lot worse. So they'd really done their homework, but in spite of doing such a thorough analysis, they were still cautious enough to make the following statement. A definitive conclusion is only going to be achieved through more data. Well, biologists love data, more data always. But also, they said, some more taxa. And they specified exactly this Xenotubelid, this new phylum, which has been found about 10 years ago, hemichordates, and more echinoderms. Well, seemingly, Bourlat et al. Must have been reading this paper because they did exactly that. They got more genes and got those three taxa exactly, a hemichordate, a Xenotubelid, and a starfish, and redid the analysis using as a core Delsuc et al.'s data set. And what they found is, miraculously, the cephalochordate was no longer a sea urchin. It became a vertebrate again with very, very compelling, a chordate with compelling results, but the tunicates remain at the base of vertebrates. So, thank goodness, deuterostome phylogeny reveals monophyletic chordates, so everybody's happy again. But the tunicates still stay in a funny place. They were also able to show that it was just adding three taxa that did the trick, because when they removed those three taxa, they were able to get Delsuc et al.'s result on the left, and then when they added them back in, they got the result that they presented in their paper. So what's going on here? Well, obviously, the fact that the data are so sensitive to taxon sampling means that the model describing the process is suspicious. If the model's good, and you have a good description of the process, and you add extra taxa, that core set of relationships should remain intact, and it should plug in somewhere. If by adding in one taxa, it sort of by remote control screws up the rest of the tree, obviously you've got something going on with the model that's not appropriate. So we wondered if it was a bad model. So Clements Lackner laboriously went through and did a partitioned Bayesian analysis under WAG with gamma with independent rates for each of the 170 sites and gets exactly the same results. So if it's a model problem, it's not one that you can fix with simple rate multiplier tailored to each gene. Could it be non-orthologous gene comparisons? Got 5 minutes, okay. Well, orthology can be a huge problem with ESTs because basically you get some little string and you blast it against another set of ESTs or against a database. You take the one that's closest to it and then you do the reciprocal, and so orthology is defined as being identified by a bidirectional hit. And if you don't get a bidirectional hit, it's called non-orthologous. Well, there's lots of situations where pairs of strings meeting the criterion for orthology are not true orthologs at all. You can have rapid evolution in one ortholog, which then renders it more dissimilar to its true ortholog in another species. And there's other situations too. So this is a problem. So we thought, well, first thing we want to do is know if the input data are reasonable. So we thought, well, we've got all of these 170 genes. What might be going on? Let's filter them and see look at the subset that actually have really bloody obvious groupings. We want insects to be monophyletic. There's a mosquito in the data set and a fruit fly. We want those two to come together. There's a couple of sea urchins. We hope that they come together. And there's a lamprey and a human, and we hope they come together. So we thought, let's ask, out of the 170 genes, which subset actually meet that criteria? So we filtered them all. Only 16 of 170 actually met that criteria, which was a bit of a surprise. So we thought, well, we'll contrast the signal of that original set of 170 genes with a filtered set of 16 to see what happens using consensus networks. And here are the results. Here's the network consensus of 170 bootstrap parsimony trees. So we bootstrapped each gene individually, got the consensus tree, and then used those as input data. And here are the results for just the subset that met our positive control criteria. And rather interestingly, the cephalochordates for that subset actually come out as the sister group to vertebrates. And here are the vertebrates, and here are the tunicates out there. So we thought, aha, we think we know what's going on. But no, we don't. Because when we went and did an amino acid likelihood of those 16 genes, we find out that we still get cephalochordates and echinoderms coming up together. This is Del Sucadal's original result. So something else is going on. There's also some other very strange groupings going on. So this is the bit I chopped out. We tried Nicolas Lartiot's cat model. That also shows sensitivity to taxon sampling. So there's still some model inadequacy. So there doesn't appear to be any sort of quick fix here. So then, what else could it be? Well, going back to sort of first principles, we know these protein coding sequences code for structures. And we know in general that structures show very limited variation among lineages, but they do show a little bit. Here's some superimposed Herudinian structures from 28 different taxa. 1 minute, okay. We also know that patterns of variability vary across sites and taxa, which results in these trees with incredible variation in branch lengths. So is it possible that what we've got going on here, I don't find it to stick, is that we've got different freedom to vary in subgroups across different, so primates have got different variation in these particular sites than do fishes. So if this is true, what we need, if these changes in freedom to vary are going to require that amino acid frequencies of mixture models are allowed to change over the tree. So what we maybe need is mixture models that better accommodate freedom to vary at sites mixed with a covarion hybrid kind of model. So some practical sort of points. The strategies that I think are probably most reasonable if we're interested in phylogenetic accuracy is, number one, and you might think this is a no-brainer, but I think it's important, is ensure that the input data meet some sort of minimum criterion that ensures orthology. Otherwise, you chaps can go blue in the face. Mathematicians can do all sorts of wonderful things, and it's just going to keep you busy. And the other thing is a very sort of practical approach is we could minimize among lineage heterogeneity by excluding those regions or genes that seem to exhibit these non-stationary covarion mixture model kind of dynamics. And so housekeeping genes that have basically the same function that are locked in over huge tracts of time might be good candidates because they've got similar constraints. Alternatively, if you're ambitious, we could try to optimize parameters on a sort of structurally informed gene-by-gene basis to accommodate context-dependent evolutionary change. Obviously, we're not going to be able to do that for hundreds of genes, maybe one or two, because that's a huge amount of work. And that's actually work that Clements Lackner is interested in doing right now. So So the bottom line is that I'm not that optimistic about these ESTs because it's very quick and it's very easy and the numbers look very big, 200 genes, 300 genes, but I think that it would be really, and also these data sets are replete with missing data. 50% of them have no entries in them because we thought it was just a missing data problem, so we sort of screened through to get rid of that and there was not much left. So as data sets include more characters, sampling variance goes down, and we're not shielded from the effects of model misclassification. So accurate estimates are likely to come from a better appreciation of transformational tendencies associated with individual sites that perhaps should be biochemically motivated. Until then, we have to sort of prop up our inadequate models with thoughtful taxon sampling. Well, that's easy to say, but some of these taxa don't exist. If you've got a bloody great rift between two very distant taxa and there's nothing in the middle, you're toast. So, and I think that, and this is an opinion, not really a fact, I think phylogenomics as currently practiced is close to the worst case scenario. We've got long sequences, ambiguous orthology, divergent taxa, and sparse taxon sampling. So with that, I'll just acknowledge Clements Lackner, who did all the hard work, and Mark Holder, who stopped me from saying things which are really crazy. Yeah.
I was wondering if there's an algorithm out there that tries to help you pick the best protein model. I don't know if you're familiar with this at all, and I don't remember the name of it, but you can give it an alignment, and it'll say you should be using MT-Rev, or you should be
Rafael
using
Sardoyas. Okay,
right.
yeah.
And I was just wondering what your thoughts are on that.
Yeah, they're all IID models.
Right.
Right, and so I think what's going on is that you've got a lot of interaction among critical places, which is fine when you've got long internal branches, but when you've got some of those really tricky little internal branches, then IID assumptions can ruin your whole day. I think that what you've got is interaction going on that might actually compromise it. But we haven't tried that, and it might be one more thing to try.
I just was wondering whether you knew if they work well or not. But
I haven't tried them, but it's a good idea. Yeah. Yeah, Andrew.
Look amongst different genes in that 170.
Yeah.
Do you find that there's some that support what you think is probably the correct topology?
Yeah. But what's interesting is I can't just pick and choose like that because whilst they might support the correct topology, they also have comb jellies grouping with fungi. So, there's no sort of perfect bullet in there. And we try to be objective by having criteria beforehand and then seeing what happens. So, yeah.
So we tried testing the adequacy of the models. So if you simulated data under, say, your WAG plus gamma, whether the pattern frequencies you see look anything like the frequencies you've seen in the data.
No, that would be a good thing to do. That would be, yeah.
Really that hash.
Right, exactly. That would be a very helpful thing to do. Clements, you have to do that.
Any further questions? Louise?
I mean, there's not only this problem where are the actually Lodermata and so on in this tree, on the Cordota side, or the, but also the active-Zoa hypothesis.
Right. That's
another side.
Yes, that's exactly right.
Did you try your methods also to check on this hypothesis?
No, I haven't had a look at that, but I think that Hervé Philippe's group and Nicolas Lartiot have had a look at that. And what's interesting is sometimes they come up with strong support for it, but then when they're trying to address a different problem, it goes away. So I don't know. So that's interesting.
More question. Did you ever try to use Adamar transformation for
I don't know.
for the data? That would be interesting, in particular for these mitochondrial genome data, I guess. If it can be done, I don't know.
Yeah.
And EST, people told me, won't be of much interest with the ultra-fast sequencing methods coming up now. So what do you think about that?
Well, I think all these methods, I'm not against ESTs, I'm just for homology, really. So,
all sequences of everything.
if you can find some way, some independent criteria. And what's interesting is that the criteria we usually use for orthology are trees, but they've substituted that for high throughput pipelines with similarity. So it's sort of one step back, really. So,
I think we should
go.
stop now.
Yeah.